Okay, here we are, and today we're going to talk about replication. Replication is taking a DNA chromosome, one of them, and making an exact copy of it through means of semi-conservative replication. The definition of semi-conservative replication is that you have one old parent strand and one new daughter strand, and they create the exact same template and DNA chromosome for both. So we are going to go through this PowerPoint together. Yay, PowerPoint. DNA replication. DNA replication is a part of the cell cycle. If you can see here, a cell spends a majority of its life in interphase. Interphase is growing, G1. The growth phase is done to make sure that it has enough cell reserves and enough proteins and mechanisms available to go into synthesis, D or S. This is DNA synthesis. This is where we actually copy the DNA to make two daughter cells. Then you have G2, which is a second growth phase to make sure the cell has enough resources available to turn into two different cells. And please see here, this indicates a checkpoint. If all the factors of G1 are not met, it rests here until it is either fixed or it undergoes apoptosis, which means that it does self-death. Cell death is what apoptosis stands for, and the cell can be told to kill itself because it is mutated. Here is also another checkpoint. This is a very important checkpoint because if the DNA was copied wrong or the DNA was massively mutated while being copied, you don't want to make daughter cells out of that DNA. And so there's another checkpoint here. And there's a checkpoint at the end of G2 or the second growth phase right before the cell splits into two daughter cells to make sure that the cell is ready to do that. Then, so DNA replication occurs in the S phase, and then the mitotic phase or mitosis is where the cell undergoes PMAT, prophase, metaphase, anaphase, telophase, and then turns into two daughter cells through cytokinesis. So here is the double helix model of DNA, discovered by Watson and Crick. No, it's really a misnomer. We've already seen the history of it anyway, but you know by now that Watson and Crick, while very lucky in determining the structure at the very end, did not understand how DNA was done until they saw Rosalind Franklin's X-ray crystallography that showed an X pattern in the DNA, and the X pattern led them to believe that the nucleotide bases were to the inside of the DNA. They had been building models to where the nucleotides were sticking out. They wrote a, very beautiful one-page paper about their discovery. That's how they published it to the world in Nature in 1953, with the mic drop ending sentence, something along the lines of, I'm sure we've already talked about it. It has not escaped our notice that the mechanism for replication, the structure of DNA implies a mechanism for replication or something like that. It was a huge mic drop for the scientific community, because not only did they prove that DNA, at that point, they had proven that DNA was the hereditary material. Remember, they used to think it was proteins because they were much more common and had many more bases. And so it made sense to them in science. And of course, if you just think about it logically, that the proteins would be the hereditary macromolecule, but DNA is. DNA codes for all those hereditary units by the arrangement of its 4 base pairs. But because it only had 4 base pairs, they thought that the structure was too simple in history to be the hereditary unit. By the way, I'm making sure I say all this for a very specific reason, wink, wink. The double helix structure of DNA is like a ladder. You have the railings of the ladder, and then you have the rungs of the ladder to the inside. The rungs being the steps. The railings are made of a phosphate sugar backbone, phosphate sugar, phosphate sugar, phosphate sugar, that run anti-parallel to each other. So here we see phosphate sugar, phosphate sugar, phosphate sugar going in the opposite direction. The DNA strands are always anti-parallel to each other. This is 5' because this is the 5th carbon in the carbon structure of this ribonucleic acid. So 1, 2, 3, 4, 5. That is the 5' carbon. And down here is the 3' carbon. 1, 2, 3. So this runs 5 to 3', and this runs 3 up here. Sorry, 1, 2, 3 to 5'. 3 to 5, 3 to 5. That's going to play a huge role in how DNA is replicated. The nucleotides are joined, these are covalently bonded. The railings are, the phosphate sugar backbones are covalently bonded. The nucleotides in the center, adenine, thymine, cytosine, guanine, are connected by hydrogen bonds. Hydrogen bonds all the way down. Now we know that one single hydrogen bond is weak. But when you have millions and millions and millions and millions of base pairs hydrogen bonded to each other, they are very strong. So something has to go along and break all those hydrogen bonds in order to separate the parent template strands to build those 2 daughter strands in. Adenine and thymine always bind together. They have 2 hydrogen bonds. Cytosine and guanine always bind together. They have 3 hydrogen bonds between them. It's always going to be 3 hydrogen bonds for cytosine and guanine. It is always going to be 2 hydrogen bonds for adenine and thymine. Remember, please, that each nucleotide, by knowing how much of any individual nucleotide you have, you can figure out the amount of the other nucleotide you have. These are Chargaff's rules, that the amount of adenine are going to equal the amount of thymine, and the amount of guanine will equal the amount of cytosine. So, for example, we'll just do something really easy here. If I had 10% cytosine in the cell, how much guanine do I have in that cell? If I have 10% cytosine, it means I have to have 10% guanine as well, for a total of 20% cytosine guanine. Okay, I have 20% cytosine guanine. That means I have 80% of my nucleotides left to discover. They have to be either adenine or thymine. If I have 80%, that means 40% is going to be adenine and 40% is going to be thymine. That's how you do those problems. Wink, wink. There is that sentence at the end of the 1953 article in Nature. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Boom, mic drop. The whole advent of genetic biology born from that single sentence. It's ridiculous. Right, so I've mentioned 5' to 3' and 3' to 5', and it is very, very important that you understand the directionality of DNA, or you're not going to understand replication at all. You need to number the carbons. It matters. Here is your nitrogen base. Here is an oxygen, and then we start from there. Here, 1 carbon, 2 carbons here, 3 carbon there, 4, and then 5. And so this is 5' to 3'. That is why it is called that directionality. This will be important. When you put a backbone of DNA together, which remember the backbone of DNA is the sugar phosphate backbone. Phosphate, sugar, right? You have to refer to the 3' and 5' ends of the DNA. The phosphate will only connect to a 3' end. Let me repeat, the phosphate will only connect to a 3' end, which means you can only add nucleotides to a 3' end, a free 3' end at that. So one that is available, that is that 3rd carbon. That is the only place you can add nucleotides. Please remember that. I keep looking at myself because I'll look at the camera from now on. Sorry, I've been looking sideways. So that last carbon, there's the 5', that last trailing, oh my God, 3' carbon is where you add the phosphate too. Sounds trivial, but this will be important. Nucleotides in the DNA backbone are bonded from the phosphate and sugar between the 3' and 4' carbons. That means it has directionality and they run opposite to each other. 5 to 3, 3 to 5. See, I wasn't lying, not lying to you. We've already talked about the bonding. I'm apparently way ahead of myself in this PowerPoint, but we bond covalently between the sugar phosphates and through hydrogen bonds between the nucleotides. So there you go. Strong, strong bonds, weak bonds individually, as we know, but strong bonds collectively. Base pairing in the DNA goes, as I've already mentioned, your 2 purines, which, remember, smaller word, bigger molecule. So the purines have 2 carbon and nitrogen rings, are adenine and guanine. And then pyrimidines, bigger word, smaller molecule. They only have 1 ring of carbon nitrogens. That is your thymine and your cytosine. And as I've already said, A and T have 2 hydrogen bonds between them, and C and G have 3 hydrogen bonds. So how does DNA replicate? It took a long time for us to figure this out. Remember the history of DNA with Mensels and Stahl and Chargaff and Hershey and Chase and Wilkins and Franklin. It took a long time for us to figure out how DNA copied. But the way it did, we finally found out, was something called semi-conservative, where each base pairing, so if you split the strands apart, each base pair that's left on a parent strand provides a template for the base pair that goes with it. And so it's semi-conservative. You see, old is dark blue, new is light blue. It's semi-conservative so that the new strand is 1/2 of a parent template, or the old DNA, and 1/2 new DNA. There it is, semi-conservative. This is a micrograph. Here, this is a micrograph. It's not colored for you guys, sorry, but it's showing a replication bubble. You see how the DNA has opened right here. But look, here's another replication bubble. And see here as well, this is another replication bubble. This is very important for you to understand that eukaryotic chromosomes have the ability to have hundreds to thousands of replication bubbles all at the same time during DNA synthesis. Not the same for prokaryotic genetic material. They typically only have 1 replication bubble. But because eukaryotes have such large amounts of DNA to copy, if we only had 1 replication bubble, it would take way too long. We'd never be able to divide our cells at a rate that is possible to maintain our structures in this life form. We would have to be much, much smaller life forms if we only had 1 replication bubble. And it would take so much energy and time to get the whole genome done with just 1. So we have hundreds to thousands. Wink, wink, please remember that. Okay, a replication bubble is coordinated by a large team of enzymes, and you're going to meet all the players of these enzymes. And a bubble will start, and we'll talk how the bubble starts in a minute. And a bubble will start, and then they form together and merge, and then you have your 2 daughter strands. Okay, so the first step of replication is unwinding the DNA. You have your follow along notes that I sent out in the packets. That's the next part of this unit that I have recorded for you on the very next thing on the module. And you will meet individually each of these players. But the first step is unwinding the DNA. We do this with the helicase enzyme. Remember, ACE already implies enzyme. So this is actually helic enzyme is what that's saying. Helicase enzyme, unwind part of the DNA helix. And so they are representing this as this little pyramid purple shape down here. And it unwinds in both directions. Now, Once you have unwound the strands of parent DNA, they will automatically re-anneal back together. So to re-anneal means they'll reform those hydrogen bonds with each other and just wind back up. You don't want that to happen because you need them to be separated to create the new DNA. So we use SSBPs, also known as single-stranded binding proteins, to stabilize the unwound parent template strands. And these are indicated by these little blue dots on either side, and they will have to be on both strands to keep stabilized. The point between unwound DNA and still wound DNA is called the replication fork. Now, let's see, do I have one? Rubber band. Oh, good, I do. Now, so now I have a rubber band, okay? I'm gonna wind and 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 wind my rubber band. This is in no way representative of how much winding is done in DNA. But if I start somewhere in the middle of this rubber band and I start to pull it apart, what happens to the ends of my replication fork right here? That's right, they get super, super overwound and tight, and then you won't be able to open the DNA anymore. It's not good. We definitely don't want that to happen to our DNA as we're trying to replicate it. So we have to have another, sorry, I'm talking too fast for myself, enzyme to stop that from happening. And what we have is something called topoisomerase. And I don't know if it's on the next slide. It's not. It's called topoisomerase. You'll meet that in the follow me notes. And what topoisomerase does, I shouldn't have unraveled it, but it goes to those ends, those very, very tight ends of the DNA, and it clips it, unwinds it, and then puts it back to just normal winding so that you don't have all that tension to where you can't open the structure anymore. Topoisomerase does that. Okay, once helicase unwinds the DNA and the single-stranded binding proteins are stabilizing the DNA, then we can start replicating. But it's not as easy as just throwing in some nucleotides and seeing what sticks. We actually have two more enzymes that have to start replication. Replication is to build the daughter strand off of this parent template strand. So this is going to be old parent DNA, and then we have new daughter DNA that's going to be built in the opposite direction, remember. So it adds complementary bases. This is DNA polymerase 3. It is super important that you know that this is 3 because in transcription, there are different polymerases. And DNA polymerase 1 does a whole different step in replication. So it is important that you know it's 3. Woo, there's DNA polymerase 3, but we're missing a step already. Sorry, DNA polymerase has no idea where to start replicating. It actually has to be bound with a promoter, and they're not going to show us that here, but it's a promoter region. And in fact, the DNA has very specific sequences for the promoter region. It's called a TATA box because it's TATA. Now it can be like hundreds of TATAs, but it's thymine, adenine, thymine, adenine, thymine, adenine, TATA. And that thymine adenine box tells RNA primers, not DNA, RNA primers where to bind. And then once the polymerase recognizes those primers are in place, then it binds and starts to add nucleotides, which means that something has to come along and erase the RNA primers and put in DNA in their place. And that's DNA polymerase 1. But we'll get there. So DNA polymerase 3, after seeing the TATA RNA primers, binds there. That's its indicator. This is where you start replicating. And then it adds bases. So if you have a C, you're going to add a G. If you have a T, you're going to add an A. And please note that the DNA polymerase is building in the 3' to 5' direction here, because you can only add on to the free 3'. You can only add on to the 3' carbon. And so if you have a guanine, you put a cytosine. If you have an adenine, you put a thymine. If I have a guanine, I'm going to add a cytosine. Okay, I'm missing something. What? Oh, the energy to bond. Right, so everything takes ATP. That's all I really have to say about that. Okay, remember that normally in cell cycle energy or energetics, when you take a phosphate off an ATP, you get an ADP, and in metabolism, we just recycle that back to ATP. But please note, once again, remember I mentioned this back in enzymes, that if you take off 2 phosphates, then you just have a regular nucleotide, an AMP, a monophosphate ribose sugar nucleic acid. When we take off 2 phosphates, we can add that into the phosphodiester covalent bond of that sugar backbone. That's where we get our energy from. Okay. And I did note to you that there is GTP for the guanine, there is TTP for the thymine, and there is CTP for the cytosine. We just don't usually use GTP, TTP, and CTP for energy currency in our cells, which is not true because sometimes we do use the GTP, but we use ATP much, much more. So the nucleotides arrive as nucleosides with their 2 extra phosphates. We use the extra phosphates as energy for the binding. There they all are. And therefore they arrive with their own energy to bind into the phosphodiester covalent backbone of the sugar phosphate backbone of DNA. They're bonded by the enzyme DNA polymerase 3. So DNA polymerase 3 is just facilitating the bonding. It's not adding its own energy to it. Only can be added, you can only add bases, once again, to that free 3' end. And so there we go. You bring in your own ATP, and you use it to that energy. In the DNA polymerase, this works its way down. Once again, I know I'm beating a dead horse at this point, but it is super important that you understand that it can only grow in the 5' to 3' direction. Why is that super important? Because only one of the daughter strands is going to be growing in the 5' to 3' direction. This is called the leading strand because it can just grow straight down. See, look at it go. Boom. However, these are anti-parallel strands, which means that the other strand is going to be growing in the 3' to 5' direction. Okay, so now we have our 5' carbon. We can't, this is not, you can't. There's no way to add this phosphate onto this carbon to make that bond. It doesn't work. There it is. That's what I was waiting for. Sorry. It doesn't work. So what do? We have to start somewhere in the middle and build backwards. And then we have to move up and start and build backwards. And then we have to move up and start and build backwards. And we call this the lagging strand because it's not going to be going as fast as the leading strand. Oh my God. But that means that we are going to have openings where there is not a bond. And something's going to have to come along and fix that opening. Is it another enzyme? It's ligase. So ligase is our gluer. If you watch the Amoeba Sisters video of a replication that I have posted, they have this cute little picture of a yellow enzyme holding a glue bottle and he's really excited. It doesn't use glue, obviously, but it does come along and fix all of these little openings that are in this lagging strand. The lagging strand is also known as Okazaki fragments because Mr. Okazaki or Dr. Okazaki, I don't really know, was the man who discovered that this was happening. He discovered the lagging strand part of the DNA synthesis. So ligase will fix all these holes, which is good. So let's look at the big picture of your leading and lagging strands. Whoa, it's going faster than I thought. This yellow guitar pick shaped doodad, who is this? He's unwinding the DNA. Good, this is he. And then these black dots now, they were blue earlier. What are these on either side of the replication fork? These are SSBPs, our single-stranded binding proteins that that make the parent strands stay apart. Remember, otherwise they would just twist right back together with their hydrogen bonds reforming, and that's called re-annealing. So 3' to 5', 5' to 3'. You'll always want to make sure you have those marked. Anytime you're doing any kind of testing that has the 2 strands in your DNA structure homework packet, every time you see it, you want to make sure the first thing you do is mark the 5' and 3' positions so that you'll know which one's leading and which one's lagging. All right, once again, this PowerPoint is missing the point that you have to have RNA primers before the DNA polymerase can bind. So just in your head, know there are RNA primers in the spot that the DNA polymerase is bound. So if this is 5 to 3 this way, that means this is 5 to 3 this way. So this is going to be our leading strand because we're going to have the free 3' carbon available to just add nucleotides to. Okay, so that's good. So it's just going to build. But here we have our 3' to 5' end. We can't do that, remember. So we have to start in the middle somewhere and build back, and then start somewhere else and build back, and start somewhere else and build back. These are the Okazaki fragments. So that is the limit of DNA polymerase 3. It can only go in the 5' to 3' direction. Why? Evolution, man. That's why. Oh, good, there's the name, Okazaki fragments. And then remember our good friend, Mr. Ligase, with the little glue bottle, he's so cute, will come along and fix those holes in that backbone and make that last covalent bond so that it is complete. The leading strand, once again, continuous synthesis, no stopping. Lagging strand, fragments that have to be joined together. All right. Once again, the very first thing you do is your primes. 3', 5', 5', 3'. We have to make that replication bubble. Remember, this is opening both here and here. You have helicase on both sides. And remember, in eukaryotes, you can have thousands of these replication bubbles happening at the same time. All right, so we're going to go both ways. They're not showing you helicase. They're not showing you the single-stranded binding proteins. And once again, they're not showing you the RNA primers. DNA polymerase is building 5 to 3 along that strand. There you go. This one is building 3 to 5. And so you have to do it in fragments. And this is what it ends up looking like. What? 5 to 3. 5 to 3, yes. Okay, so eventually what they're trying to show you is that on either side of a replication bubble, because of the anti-paralleledness of it all, you will have a 5 to 3 and then Okazaki fragments. And then you will have, once again, a 5 to 3 and then the Okazaki fragments as you open up that bubble more and more. Because the DNA polymerase 3 just binds in the center and starts. So you have to have DNA polymerase come back and bind in the parts of the replication bubble that were not available at the very beginning to fix that problem. Right, so we already know the limit of the DNA polymerase 3 is that it can only build 3 to 5. Oh, look, finally, primers. We're going to talk about the RNA primers. Wow. And so here it's showing you as a red box the RNA primers that are having to bind before DNA polymerase can bind. So far, just in general, while we're thinking, helicase, single-stranded binding proteins. Helicase unwinds. Farther down the DNA, topoisomerase is undoing the extra unwinding that is happening. Okay, RNA primase, ACE enzyme, DNA polymerase 3, build. Ligase comes along and fix the holes in between the Okazaki fragments. So there's a primer, there's a primer. Yay, the primers have to bind first, and they are a starter sequence. Remember the ta-ta box for DNA polymerase. Here is DNA polymerase 1. I did mention him earlier. 1, not 3. Different polymerase, different enzyme. Named for the order that they were discovered. DNA polymerase 2 was discovered in prokaryotes. He's prokaryotic, so that's where he is if you want to know where he is. DNA polymerase, ligase has nothing to do with the RNAs, but our DNA, our genome is not DNA with little bits of RNA in it, it's all DNA. And that is the job of DNA polymerase 1, is to come back and delete these RNAs and fill them in with DNA. Once again, why such a convoluted system? Evolution. Oh my gosh. There. So DNA polymerase 1 removes sections of RNA primer and replaces with DNA nucleotides. But DNA polymerase 1 can still only bind in the 5' to 3' direction because it's a polymerase. Okay, I've already said that. So they want to know, what's going to happen here? What's happening now? We lose, at the very end of our DNA, we lose bases because we cannot build 3 to 5 at this very, very tip and replace those RNA primers. So our chromosomes get shorter with each replication. Along with oxygen consumption, which is toxic to us, along with free radicals being formed in our body and turning into hydrogen peroxide and hurting us, or just being extra protons and hurting us, our chromosomes get shorter with each replication. These are all hypotheses of why we age. Now, we have an enzyme that makes our aging very slow. It is called telomerase. Those very, very ends of the chromosomes actually end up being nonsense DNA because of this phenomenon, because we lose them during replication. So it's all nonsense DNA. It's DNA that doesn't code for any proteins or anything that we don't use in our bodies. And that's what gets shorter and shorter. Now, there are genetic mutations that people are born with that they don't have telomerase, and they age very, very quickly. A 10-year-old can look like a 90-year-old type of thing. But we have, most of us have telomerase that goes back and puts in more of the nonsense DNA, so that even though they do shorten every time, they don't go as short as they could be. Eventually though, yeah, they get short enough to start causing mutations in the chromosomes, which is why ladies specifically, we have like a age on our eggs and our ovary, because remember, you're born with every egg that you can ever create a baby with, and they as they multiply and get older, it's not anymore. So telomerase, these are that nonsense DNA. Telomeres are what the nonsense DNA is called, that cap our chromosomes. And then you have telomerase that extends them with every replication. It has different level of activity in different cells. And so in terms of activity level, you will find that telomerase is high in stem cells. Now remember, stem cells are cells that haven't decided quite what cell they want to be yet. And so they can go in many different directions, and cancers. And the reason why for that is very different. Cancers, they're mutated. Cancers can live forever. We have HeLa cells that we do a whole bunch of therapeutic testing with. HeLa cells come from Helen, oh, I should know her last name. Anyway, without permission, they harvested her cancer from her and kept it growing in a lab without her family's permission after she passed away. And Henrietta Lacks, whoa, Henrietta Lacks is her name. Please don't forget the name Henrietta Lacks, because she is unknowingly one of the reasons that we have so many wonderful and chemical therapeutic drugs on the market for cancer. But Henrietta Lacks had, I don't remember what type of cancer, I'm sorry, it might have been ovarian, and she died from it. But in the course of treating her, they harvested the cancer from her. They got a tumor from her, and they kept it alive. The scientists kept it alive in a lab without her or her family's permission. And they grew cell lines from her tumor that they then used and shipped all over. She's everywhere. Henrietta Lacks, her cancer is still alive. Cancer can live forever because the telomerase activity in this cancer is so high, because they're so highly mutated. Now, that's not the only reason cancer lives forever, but it's one of them. And stem cells, because they have so many different pathways they could choose, you don't want those mutating. And then one time, I think our final time here is our replication fork. Here is helicase unwinding the DNA. Please remember that somewhere down the strand, topoisomerase is undoing the tension. You have your single-stranded binding proteins stabilizing the parent templates, sorry. You have primase that is setting these RNA primers in place. The RNA primers tell DNA polymerase where to start. And you see multiple DNA polymerase working. Well, for the Okazaki fragments, you have to have ligase binding that final covalent bond together, but then you also have to have DNA polymerase 1 here to remove the RNA fragment. And you know now about the leading strand and the lagging strand. DNA polymerase 3 is a wonderful enzyme. We have millions of copies of it in every single nucleus in every single cell. It can base pair up to 1,000 bases per second. And of course, it's our main DNA builder. Unfortunately, that means it also has a high mutation rate. We actually, there's like 1 in 1,000, I think we mutate 1 in 1,000. It's pretty high for our mutation rates, but we have something to fix that. And then and that is DNA polymerase 1, sorry. And he goes much slower. He does 20 bases per second, and he edits and repairs DNA polymerase 3's base pairing and primer removal. We've already seen that. So, good. And Arthur Kornberg is who discovered one. And Thomas Kornberg, I don't know, oh, he's not dead yet, discovered DNA polymerase 3. So, I told you 1,000 bases per second is lots of typos. So DNA polymerase 1 comes along and proofreads and corrects and repairs mismatched bases and removes abnormal bases and reduces the error rate from 1 in 10,000, sorry, 10,000, not 1,000, to 1 in 100 million bases. Errors still occur, but so much of our DNA is silent. So much of our DNA we don't use to code for proteins. Typically doesn't matter that the error has occurred. So that's good. It takes E. coli approximately 1 hour. It's really about 45 minutes in optimal conditions to copy 5 million base pairs in its single chromosome. Human cells can copy its 6 billion bases and divide into daughter cells in only a few hours. It's remarkably accurate. Only approximately 1 error per 100 million bases, 30 errors per cell cycle. So it does not look like a cartoon with 2 beautiful lines and a nice bubble in the middle. This is kind of what it looks like. Here is a single strand. This is a circular chromosome, so it lets us know we're in a prokaryote. No, it doesn't, because we have multiple. It doesn't matter. And then we have here, you can see a fork here and here. And this is a replication bubble. They have it colored in for you over here. And then here is a fork, And there's a fork, so there's a second replication bubble. Here is a, nope, that's just a loop. Here is a third replication bubble here. And I gotta find it. Here is the fourth. Here-ish. Something. Anyway, there's a fourth one there, if you can see it. Good. Woo! Any questions? Oh, this is a much nicer. Look at them. Look at that nice 3D models of enzymes. If you have questions, rewatch this. Or if you think your questions could be answered by doing the follow along notes with this that I sent out to you called DNA replication. Sorry, they're sticking together. Oh my. Anyway, it's 3 pages long, and what we're gonna do, we're gonna cut out the boxes and tape them together, and I'll show you what that looks like at the beginning of the follow along notes for DNA replication. So, excellent. Thank you, and goodbye.